Good. So next, let's talk about how to determine apoptosis. Because if you remember, I already explained to you that cleavage fragments of caspases are not sufficient to tell you that apoptosis is occurring or still occurring. Now to summarize, so far I have introduced the Western blots of caspase cleavage with its limitation. We have talked about cytochrome C release and the loss of mitochondrial membrane potential. Cytochrome C release and MMP loss are upstream of caspase 9 and therefore upstream of caspase 3. That means both events could have happened and you still don't get caspase activation because of XIAP. So those are all good measurements. They are solid measurements done in the literature. However, they are probably not sufficient. Or let's put it another way. If I write a paper, I try to get away with using one method. If I'm a reviewer, I want to see at least two methods. So what else could we look at? There's tunnel staining. Tunnel staining is often used in histological slides, where you basically label damage of DNA. So if the DNA begins to get fragmented, you label it and it's therefore highlighted, as you can see with the little red arrows. Those indicate cells which have a bad DNA damage, leading to probably the fragmentation of DNA being a hallmark of apoptosis. These nicks in the DNA, thus damage in DNA, can also occur when a cell is dividing. So the end of chromosomes can be also positive in tunnel staining. So again, tunnel staining is a very good indication, but probably not sufficient. It is also something which you should have a student that you don't like do, because basically it involves counting lots and lots of dots. And then you see lots and lots of dots for days without end. You, however, can do this computer guided these days. You can do also something similar by staining your histological slides for active caspase 3. So there's an antibody which just detects not the pro form, that wouldn't tell you anything, but the cleaved form. Again, that involves lots of counting. It is a very slow process. A very quick process using a 96 well-played format is the so-called MTT test, where you have a water-soluble yellow dye that gets reduced to a purple, brownish, blackish dye that is water insoluble. So basically you could use lots of different concentrations and lots of different cells, and you'll get a hell of a lot of information. You also only need minimum amounts of substance you want to test or of cells. So here you basically have every black dot in here is a cell that is reducing the yellow substance to the purple dye. It used to be assumed that this process occurs in the mitochondria. So one assumed the MTT test was an indicator for mitochondrial activity, which is of course closely linked with apoptosis, where you have the loss of MMP and the inactivation of mitochondria. However, it is now known that these reduction processes also occur in or at the ER. So you have to remember this assay basically only tells you the metabolic activity of a cell population. It does not tell you whether your treatment, for example, reduced the amount of cells by inducing apoptosis, whether your treatment reduced the amount of cells by stopping proliferation, or whether the treatment reduced the amount of cell, didn't reduce the amount of cell, but sort of forced the cells to hibernate to reduce their metabolic activity. So the MTT is a very good assay to give you an idea if you want to test a substance whether it affects your cells, but it's not a particularly good assay if you want to exactly know what's happening. Now I always like to use the FACS because that allows you to measure thousands and thousands and thousands of cells in a few seconds or minutes. And basically what you can do if you propydium iodide stain the cells, that is, if you permeabilize the membrane and then force the propydium iodide into the cell, you can stain the DNA. And you get a very good idea if a cell has fragmented DNA because the small fragments get chucked out of the cell. So if a cell has less than G1 amount of DNA, when we get to that, these cells have fragmented DNA and are probably apoptotic. Now that machine then calculates the amount or the percentage of apoptotic cells in your population. It saves you a lot of work. So basically we have here a nice example from my younger days, and it goes hand in hand with the lecture you had on cell cycle progression. If you remember your lecture, you have cells which are in G0 or G1. Then you have cells which are in S phase, where the DNA is synthesized, and you have cells which are in G2, where the cells have the double amount of DNA and prepare to divide. Now, if you activate an oncogene, in this case VSARC, first oncogene ever to be discovered, 1911, by Peyton Roos, what you get is what you frequently get with oncogenes is that they drive the cells into proliferation. So you get less cells in G0 and you get lots and lots of cells which are driven through the cell cycle. The very classical example for this is the c-Myc oncogene. Now that normally causes a huge amount of stress, and a normal non-transformed cell would die. Now these are chick embryo fibroblasts, normal non-transformed cells. However, they survive because they express a protein called FAK, focal adhesion kinase. Now FAK is completely, well, not completely, but mainly unimportant. So if you have normal chick embryo fibroblasts, which you have here, which express a version of FAK which is not active, or which can't be activated via phosphorylation, to be precise, then those cells are fine, nothing happens. However, if you then combine the activity of the oncogene with the inactivity of FAK, then you get a huge amount of DNA fragmentation. So you drive the cells into apoptosis, as analyzed here, where you get an apoptotic index of 20-odd%. Okay? I find that very exciting. A different method also uses propydium iodide, but importantly does not damage the cell membrane. So propydium iodide can only go into the cell if the cell membrane is made leaky, which we do in the so-called Nicoletti protocol by adding triton X. In this double staining protocol, we don't make the cells leaky. What we do instead is we give pyridium iodide to them and we give annexin 5 to them. Annexin 5, remember, binds PS, early stage apoptosis. So what you have here is unstained cells, cells which are not positive for PI and not positive for annexin 5. They are healthy cells, completely intact. What you have here are cells which are positive for PI and annexin 5. That means these cells have not only induced apoptosis, but also are at the stage of apoptosis which we call secondary necrosis. That's all in the lecture, where the membrane becomes leaky. Also implying that these are the cells which are early stage apoptotic. They are just going into apoptosis. Okay? So you have a progression from here to here to here. Which brings us to the end of our lecture, which I hope you have enjoyed, and the key points you should have taken home. I've hopefully showed you convincingly that cells can die via many different routes. There are many different forms of programmed cell death. Apoptosis is one of them, and in my not so humble opinion, probably the most important form. We have discussed that apoptosis occurs naturally in the human body or in any multicellular organism, even C. elegans, which gave a Nobel Prize a couple of years ago, and it's an important part of normal development and of everyday occurrences. Apoptosis can be induced in normal cells if something has gone wrong, if you have exposed your cells to cigarette smoke, too much sunlight, etc. And before these cells change, they are induced to die. This suicide program is extremely tightly regulated because you don't want too much cell death and you don't want too little cell death. And you want to have control over every stage of the process in case something occurs which can stop it. The key players, the key mediators of apoptosis are caspases. They're the most important ones you have to keep your eye on. And there are different forms of how to measure apoptosis induction. Quite a few of them involve caspase activity or the results of caspase activity. So if apoptosis is such a great mechanism, such an important mechanism which is so tightly regulated, how come that we can't kill the bloody cancer cells? And that we will discuss in the next part of our lecture. So that was the first part of our lecture course on apoptosis. In that part we discussed the basic underlying mechanisms of apoptosis. In the next part we will talk about how apoptosis relates specifically to cancer. So I hope you enjoyed that part, and off we go to the next part.